side. And this is a clumsy way of putting it, but I think what genomics has the opportunity to do is to redefine the taxonomy of human disease. Redefine what we think about as Alzheimer's. It's not one disease. There is no Alzheimer's that's everything to everyone, just like there is no breast cancer that's everything to. There's 80 different histological subtypes of breast cancer alone, demanding their own different treatments. And yet we go in and say, we're going to make a drug for Alzheimer's disease. One? Really? How about we think about it as it's different? You get to a clinical spectrum that looks like Alzheimer's from many different ways that may be treated in many different ways. So to me, that's the opportunity genetics has. Let's reduce that to the form that is manageable, that has always been the promise of genomics, and it's precision medicine. Let's think about the diseases as collections of related diseases, but with different pathways and probably different treatment paradigms to impact those pathways.
This is the Helix Exchange, the first podcast about the business of genomics.
Welcome to the Helix Exchange. Today we're joined by one of the most influential leaders in modern genomics and translational biology. Dr. Lon Cardon is the president and CEO of the Jackson Laboratory, known to most simply as JAX. And JAX is a global powerhouse in genetics, model systems, and biomedical innovation. And with their recent acquisition of the New York Stem Cell Foundation, JAX now sits at the center of what we at GMG call the genomics economy, where engineered biology, data, and DNA-driven design create new products, new platforms, and even new markets. So Lon, welcome to the Helix Exchange. What an honor to have you with us today. I've really been looking forward to our conversation.
Yeah, as have I. Thanks for inviting me. I appreciate it.
Yeah, so I want to start with our signature question, one we ask all of our guests. So Lon, what's in your DNA? When you look across your career that spans, of course, periods in academia, global pharma, and now JAX, what would you say is in your DNA? What's the through line that's kept your interest in genetics for all this time?
Well, it is true. I have crossed over from industry to academia and further afield a number of times throughout my career. I feel like I'm really fortunate, and my generation is really fortunate, where we can think about genetics and genomics today as this mature field where we have movies made about it and we have fantastic, amazing, technologies that we never even anticipated, like gene editing or stem cells or DNA sequencing at population levels. None of us thought about that. That's where we are today. But it wasn't that long ago. We didn't have any of that. We didn't even have genetic markers. And that's in my generation of time, where the best we could do is try to infer whether something was heritable or not, whether it had a heritable basis. And maybe we compared identical twins who share all their genes with non-identical twins, or adoptive families that don't share any genetics and things, and just try to infer, is that trait even heritable? And we've moved from that to where we are today, which is a pretty phenomenal advance in 25, a little bit more, maybe 30 years. It's just a privilege to be here. So for me, I only ever had, even though I've done all kinds of different things with my career, I only ever had two goals, and they guided me through all the decisions that I made. And the first one was the one I thought we would set on forever in the field was, can we find genes that cause human disease? Can we find them? Are they there? And the second one was, if we can be lucky enough to find those genes, can we do something with it to improve human health, to advance biomedicine as a whole? That's it. And it involves all parties. And that's what I've learned. It takes a village. You need the academic side. You need the industry side. You need money. You need society. We've got drug pricing creeps in. It's all there. So that's my story and what drove me here.
Interesting. Well, so I want to talk a little bit about JAX. Our audience are leaders, of course, in business, finance, and many of them in Fortune 500. So I would like you to explain to them what JAX is at a business and platform level, maybe your elevator pitch.
Yeah. So if you're in the genomics domain, you'll know of JAX. So I won't answer the question for those of you that already know. Most others, the Jackson Lab is about the most powerful research institute you've never heard of. And it started in 1929, and the Jackson Lab, or JAX as we all call it, invented the use of laboratory mice to study human diseases, specifically cancer, because the recognition was we can't really disentangle human diseases in humans, neither ethically nor technically. So we need models to look at them that somewhat resemble humans. And there was a recognition back in the 20s that mice actually grow tumors that kind of look like human tumors. And so it started the field of using animal mice to study human diseases. The ambition back then was, could we develop different strains, different types of mice with different diseases or different presumed genetic backgrounds? We didn't know DNA at that time, hadn't even discovered it until 1953. The goal was 50. Could we generate 50? And today we have, the Jackson Lab alone has more than 14,000.
Oh, wow.
We work with 2,800 institutions across the world, 68 countries. We're the cornerstone of biomedicine and the animal usage. We have our own research labs that do a lot of basic discovery, but we enable basic discovery from academic research right through to pharma across the globe.
Interesting. Well, you mentioned the mouse model, and obviously that's been foundational to drug development, and of course to modern biology. Really, such an important advance. I wonder, as you see genomics and organoids and AI advancing, is there a future where the mouse model becomes a little less central, or does it remain indispensable, do you think, to your work?
Yeah. I get asked this virtually every day.
Okay.
Are the days of animal models in general, are the days of the mouse numbered? And I have a strong perspective on this because I've lived on both sides. I lived and continue to live a lot of my life as a researcher, a user of all the tools I can get my hands on, including animal models. And I've lived on the industry side as a consumer of those type of things, where sometimes one's very frustrated because the answers one gets in an animal model don't reflect what ultimately happens in humans. And that's a frustration people get. There's a famous statistician who had a great quote that said, all models are wrong, some are useful. And that applies to all of this. And sometimes the animals, mice are not furry little humans. Sometimes they're remarkably similar to humans and they give some great insights, but sometimes they don't generalize to humans. And our view at JAX is that the usage of mice may and probably should change.
Mhm.
And certainly we should use the right tools for the job. Some of those 14,000 strains are relevant for some disease questions and others are not. Let's do better at picking the right ones for the right diseases. We don't do that particularly well. We use the same ones no matter what question we're asking. We tend to as a community. But let's augment those. The cell models are fantastic. AI, the potential, a lot of hype in AI in biomedicine right now, but the opportunity is great. And we need these new hypotheses that are coming out of nonlinear thinking of just one step in front of the other, which is the way biomedicine has gone thus far. So personally, from a drug discovery perspective, I do not see the days of animal models as ending. I think it would be, at least today, it would be irresponsible to put a investigational drug in humans without having tested it in some full system, where if you're targeting, for example, the heart, you want to know if you've got a problem with other organs. And you're just not going to get that today from AI, and nor are you going to get that in specific cellular models that reflect a single organ or even communication between tissues.
Well, I think people appreciate that. It certainly allays a lot of fears to know that it's been through a live system. Interesting. Well, that brings me to the present and one of the biggest strategic moves that JAX has made in its history. I'd love to talk about your acquisition of the New York Stem Cell Foundation, why it was strategically important, and why now.
Yeah, I'll give you my own perspective, and then I need to take it back because maybe I was even late to the party. So I joined JAX just 4 years ago now, as 4 years ago this week, in fact, as we do this
Congratulations.
podcast. And I came with the idea, I came straight from industry, and I came with the idea we need more than animals. We need to augment those with all the latest tools that we have today. And the reason for that is a big picture one. In drug discovery, if we're thinking about the use of genomics to enhance treatments, diagnoses, prognostics of human disease, then we need all the tools at our disposal. And we have a problem that has persisted for decades that the drug discovery industry, pharmaceutical industry, calls the Valley of Death.
Right.
And the Valley of Death is, some of your viewers may know this, not everyone probably will. You have basic science over here that tries to understand human physiology, human biology, and they do remarkable things. My first career ambition was, can we find the genes that cause human disease? The field has knocked that out of the park. It's been phenomenal success in identifying. A few years ago, someone did a review. Every week, 3 to 5 new genes for rare diseases are identified every week. So really good on that. But we haven't translated that so well into the other side of the Valley of Death, which is when you get into pharmaceutical development, drug discovery, and so on. The middle of that is where we have the opportunity to shrink. Drugs cost a lot. There are estimates that are $2 to $4 billion because you've got to account for all this attrition,
Wow.
that all the failures that go along with this. I knew a lot of people in pharma who had worked, brilliant people who had worked their entire careers and never touched a drug that made it to market.
Oh, wow. After
That's
billions.
tough. That is a tough industry. And so someone has to pay for all those failures. And so the way that gets amortized is people say, okay, the successful drugs cost this much money. They don't individually, but in order to get there, you have to cover all these failures. What if we could reduce those failures? What if we could reduce the time to those failures? Because it's a 15-year process now. So instead of being $2 to $4 billion in 15 years, what if it was, a tenth or a fraction, whatever, a small fraction of that, and a couple of years or something like that? That's where we work. And that's why I was really excited to bring together the mouse with cells. Let's get AI in there and drive these things together, better predict, however we can get there, better predict what's going to happen in humans using all the tools at our disposal to increase the probability of success, reduce the costs. Ultimately, you could talk about potential for reduced drug pricing, more accelerated timelines to getting. So the JAX opportunity is with NICEP is fantastic. We're bringing together cells with the mice. And that's what I think the whole community needs. Now, I thought I was really onto something here because I was coming from industry and I had all these ideas about bringing these pieces together. But actually, the communities have been together for a very long time. I didn't know this at all, but the term pluripotent stem cell in its first usage was coined by a Jackson Laboratory investigator in the 1940s.
Oh, wow.
Yeah. So the fields came together as human development and human and embryonic development turned into induced pluripotent stem cells. JAX had nothing to do with the Nobel Prize that was won in 2012. So the fields bifurcated, but they've always been together and they belong together. They're complementary.
I think we need to tell our viewers what pluripotent stem cells are because they're super cool.
Oh my goodness! This is rocket science, and you're talking to a statistical geneticist who's going to massacre the technology behind it. But what you can do today with pluripotent stem cells is you can take a piece of your skin or blood, or my skin or blood, and which is all developed and it has all your genes in it, the things you were born with. It has your full complement of DNA. Every cell will have that, and you can magically convert that into its primordial state before it became blood or skin, and then advance it into any tissue you want. So you end up taking a piece of skin into a little mini beating heart or a neuron or a bit of your liver. It's remarkable. And you can do this at scale and robustly and systematically. And so induced means you can induce a cell to become something else. And pluripotent means it has the opportunity to become anything. You want to turn it into a little beating? It is really rocket science. So the beauty of this is you can take these cells that have all your DNA, all the good and the bad things about your DNA, and if I want to study your heart, then I can turn it into a little, some heart muscle and study your DNA in your heart, and that's great. I can get an answer. And ultimately, I'm doing a drug discovery program. Maybe I do some molecule screening. I'm onto something. I think it's really good. Ultimately, I still need to test that in a full organism that has all the tissues. I stick that into the appropriate mouse that I may have even humanized with your DNA as well.
Right.
And you can test it much faster in cells that have that belong to the person of interest with the disease of interest. You get it through the mouse, you increase your chances of success. That's the dream.
Wow. So the acquisition then of, you say NICEF?
We say, yeah, New York Stem Cell Foundation is when, and the acronym is NYSCF. So
Okay.
NICEF.
NICEF. So, I guess that brings a serious acceleration to Jack's vision, with their stem cell automation and patient-derived organoids. So how do you see these capabilities showing up in what Jax can offer to its customers, its industry partners, and how quickly do you think that these changes or improvements are going to roll out?
Yeah. So both fields exist already. There are many groups out there and a number of biotech. There's a lot of money being funneled into the stem cell field, as you can imagine, because the promise is so great and the technology is advancing so fast. And that's great. And we want to see that happen. Where Jax has always occupied is at the sort of the core. We set the standards for mice on reproducibility, on sustainability. Remember COVID days when we were always chasing the mutations? The virus would mutate and we needed a new version of the vaccine because the virus mutated. Well, that happens. Jackson Lab for the mice set the standard. So the mice you study today are genetically the same as the mice someone studied 10 years ago or that someone studies across the world. That doesn't exist yet in the stem cell world. We need to set the standards for that. For reproducibility is everything in science. We've got to have different labs studying the same material. So we want to bring that together, and we want to be the sort of source for all of those individuals and those companies that are developing really exciting, shiny technologies to differentiate those cells into really exciting heart tissue or brain tissue or something else.
Wow. Well, the acquisition, at least from our perspective at GMG, is inside of a much bigger shift that we're tracking, and that's where you start to look at genomics as a platform technology, the biological equivalent, if you will, to semiconductors as they were to in the digital economy. So, there were the advent of chips enabled everything from our smartphones to cloud computing. And we see genomics becoming the infrastructure for not just new therapeutics, medicines, diagnostics, and so on, but also for new materials, even new food systems, new consumer products, everything again. So does that framing resonate with you, Lon? Are you seeing some of this across your research and industry and the work happening at Jax?
Yeah, I see some of that. I can't really speak because of lack of expertise and exposure to the agriculture side, or maybe some of the animal breeding side. Although I think the opportunities are even the ones we can see today are obvious and terrific in that area and going very well. The opportunity that I see because I work in the biomedical domain. I think there's a fantastic opportunity because you can put it under organs and tissues. You can put it under regenerative medicine. To me, it's all under the personalized medicine rubric. All those fall under personalized medicine. And I think we have these really exquisite molecular tools. We can DNA sequence. There's nothing more fundamental than DNA. That's what caught me. What captured me early on in my career is, wow, that's the most reductionist form. Once I know that, proteins, RNA comes from that, the proteins come from that, the environment interacts with that. And so let's start there. So I understand where you're starting from there. So we have these exquisite tools that we can measure, like pluripotent stem cells and like gene editing. We've heard of CRISPR and there are other technologies there too. But we marry those up with really clumsy clinical phenotypes that we've been measuring for 100 years. Glucose. This week we learned metformin. It's been around for 60 years. It's a diabetes drug, may have a mechanism of action that we never even thought of.
Oh, really?
Hundreds of millions of people are on metformin.
Can you share
we
what
didn't
that
really
is?
know how it worked.
Huh.
And at least that's where it's. So there's so much to learn on the the clinical side, the phenotype side. And this is a clumsy way of putting it, but I think what genomics has the opportunity to do is to redefine the taxonomy of human disease. Redefine what we think about as Alzheimer's. It's not one disease. There is no Alzheimer's that's everything to everyone, just like there is no breast cancer that's everything to. There's 80 different histological subtypes of breast cancer alone, demanding their own different treatments. And yet we go in and say, we're going to make a drug for Alzheimer's disease. One? Really? How about we think about it as it's different. You get to a clinical spectrum that looks like Alzheimer's from many different ways that may be treated in many different ways. So to me, that's the opportunity genetics has. Let's reduce that to the form that is manageable. That has always been the promise of genomics, and it's precision medicine. Let's think about the diseases as collections of related diseases, but with different pathways and probably different treatment paradigms to impact those pathways.
So interesting. So that science you're talking about, genomics, do you see everything that you just discussed really moving and changing commercial infrastructure? And do you see industries being able to rely on JAKs in a new way now that you've been able to identify so much? You mentioned in cancer that, of course, there are so many different types that are all lumped into one. And back to your original hypothesis, or not hypothesis, but your original statement about personalized medicine. I guess I'm really wondering, how does the science move the commercial side? How can we move faster, and where does JAKs fit in that shift?
Yeah, those are a collection of really, really good questions for us right now. I do believe that if we can execute well on standardizing the scaled stem cell model and linking that as seamlessly as possible with animal models, that industry will follow, because people, big pharma, most everyone already use cells and they already use animal models. Advancing those and bringing them together will not be a big charge. So that's where I really see JAKs in the near future, is let's step into that space and be the go-to provider and partner for the entire community. That part, I think, is up to execution, and I'm excited about that future and getting there. The uptake of that more broadly for industry and for pharma, that redefinition of taxonomy of human diseases, you're still going to see, with good reason, good capitalistic reason, pharma wants to go find those drugs that are going to be blockbusters. I mean, who wouldn't want a GLP-1, right? Immuno-oncology, changing the game. These are really, really big drugs and very lucrative, and you want to get on those. That's very different from starting with something that is a subset of a subset of a subset of Alzheimer's and going further. So I think what we need to do is show that this is the way biology works for some of these, right? You want to tackle Alzheimer's as a whole, you might not find a GLP-1 equivalent. You really want to do it, you're going to have to do it in a way that is best for the patient population you're looking at. That's a harder problem than just executing on a technology. So that, I think we need to show that is the way and I think the field is showing that is the way. Cancer is leading the way. And look at all the subtypes of cancer that we think about now. Look how many, there are more than 20,000 clinical trials ongoing at any given time for cancers alone in the world.
Wow.
So you think they haven't thought of different subtypes of disease? They're doing that, they're living that. And I do think that's the next hurdle for genomics, is really to help and advance the realization and the progression of that realization to better treatments and diagnostics.
Yeah, so coming from the science, from your perspective, what is closest to becoming a commercial tool or a product, maybe even a partnership? Is there a specific lab, for example, at JAKs that's really doing a good job or telling this story well?
Where I see the greatest opportunity, in fact, there was a study. We were part of it, but like most science right now, it takes a village, and there's many collaborations. This one came out about. It was published just a few months ago, and it goes by the name of the scientific community calls it the baby KJ story.
Okay.
And it goes like this. So a newborn was diagnosed with a rare disease shortly after birth. And it was one of those real sad sentences for the parents of that newborn that says, enjoy the time with your child because you don't have a lot of it. and there's no treatment, and it's not going to look good. So from that time of diagnosis to the time of having a personalized treatment for that child that was made specifically for that child with their DNA mutations, 7 months. There's a drug in that baby right now doing well. 7 months. So we go from $4 billion in 15 years to a group of really, really clever clinicians and scientists in gene editing and the mouse and cells to 7 months. How about that for a platform for a future as we go forward? And then we build out from unique, just your mutations to those which affect a few more people, 10 people, 20 people, 100 people, 1,000 people. That I would have never seen coming. And that's a future I'm just delighted to be part of.
Yeah, and I've heard something about California testing all newborns at birth and kind of looking at their a full genetic sequence. Do you know anything about that?
Well, so there have been various calls for newborn screening, newborn sequencing for a number of years now. The UK actually announced they were going to do it. Gosh, it must be 7 or 8 years ago, and they've got part of that off the ground, but they haven't done it at a population level. I think it's a matter of time. It makes sense for us because what we call the DNA you're born with doesn't change. So you only need to measure it once. It's with you. It's in your record.
Yeah.
And so you've got it the whole time, and the cost of generating that information has got cheaper and cheaper and cheaper.
Right.
So I think it's a matter of time where we, just like we do the heel pricks for babies,
Right.
and you stick it on the Guthrie card,
Right.
we get the DNA sequencing, and it's there in our record, and we can see, because not all diseases are like baby KJ and they show up at birth. What about even other rare diseases? Huntington's disease comes on at 55, 60 years old, but the signals are there at birth.
Right.
And so having that, I think that's really exciting. And I just think that's a matter of time and economics to get the uptake on that.
Yeah, and I'm thinking about pharmacogenomics. Would you be able to pick up from that newborn screening? Would you be able to pick up what therapeutics, what your body would respond to?
Some that is the field of pharmacogenomics, and that's been promised for a long time. Certainly some side effects and some toxicity signals you could pick up on the germline for sure. As we move to these more precision medicines, I think you'll see a lot more of that, because there's almost a truism in rare diseases is once a drug it's not always the case, but it's almost always the case. Once a drug is built for a disease, however rare, you subsequently find more cases out there. So there's a lot of undiagnosed cases out there, or misdiagnosed as something else. So we would find those. If the sequencing was done already, you'd find those right off the bat. And what a treat for those parents, where
Right.
they not only get news that, spend the time with your child, but hey, there's a drug out there.
Yeah.
And it might work in your child. What a treat. Huh?
Yeah. Well, getting back to JAX, now that you've got these new capabilities, who do you think should be knocking on the door of JAX now to say that they'd like to collaborate? Are you seeing an interest from new and different players?
Well, as I said before, we work with a lot of players across the academic and the pharmaceutical industry already from the mouse. What we're already seeing, though, is, this is just early days. We only just announced this relationship a few months ago, but there's real interest in putting together the mouse and the cells. And so I see already big pharma relationships. That's where I think we can make an impact right away. And we can help evolve what questions we need to be asked and how to ask those questions. I think it's in their interest to do that. I think it's in our interest to refine that. We need them. I think different questions get asked in pharma than are asked in academia for basic science. So I think pharma and the biotech industries will be amongst the first. The academic side are already going forward. So that's just moving the train further down the tracks.
So, Lon, the topic of the year, AI. How's it changing things for JAX in disease modeling, validating targets, and scaling stem cells?
We're all excited about AI. It burst on the scene. There's no question it's changing things. It works best today with problems we already know how to solve. It does them better.
Mm-hmm.
The challenge with biomedicine is we don't know the answer to a lot of these problems. So training those with real answers we don't know yet. So I think on the discovery side of science, we've got a little work to do with AI. And so I don't think it's going to replace, personally, maybe I'm a Luddite, but I don't think it fully replaces humans in interrogation and the type of sequence of questions that we do in science. But man, does it make it easier, and it's going to speed it up. And for what we're doing to link cells and mice, I think it's going to open up the new hypotheses, the way we link them, the way we think of them together. So really, really exciting, but we have to keep our eyes open too, because not all parts of AI are ready today.
Right. So a speed up of things for sure. And I know computationally too, there's some really complex math when you get into modeling disease. So I'm sure that that's an aspect of the AI revolution for you too.
Absolutely.
Well, I want to switch over to GMG, Genomics Media Group. We are looking at this genomics economy that I was talking about across 6 core sectors that we really think are going to move in the next 5 years. Of course, Jax touches most of them because in your definition of personalized medicine, our sectors are sort of all named, apart from the ones that you mentioned earlier are hard for you to comment on, plants and animals. But really looking at personalized medicine, regenerative medicine, organs, organoids, tissue engineering, things like that. I mean, our audience really values the clarity that you're going to be able to give to this in terms of what is still hyped, what is really possible, and maybe you can opine on what we've overlooked.
Mm-hmm. Well, I wish I knew all the things that we've overlooked. One of the beauties of science is it often takes you in a direction that you would have never anticipated, and genomics is absolutely no different. And I'm sure that the 6 platforms or the 6 areas that you've identified will encompass many things, and you'll miss some others because that's just the nature. I think one of the beauties I have seen in genomics, and I'm sure it's not unique to genomics, is this cycle of discovery, basic biology discovery, demanding better tools and platforms. The tools get built, which then enable more refined discoveries. The discoveries ask for better tools, and you see it again and again and again. DNA sequencing or gene editing or all the cell work that we're doing and the robotics that come behind it. That's really cool from a platform perspective, and it's enabling new applications outside. So, and that's already real. That economy is already existing. And the pharmaceutical, Regeneron was built on genetics and genomics. Vertex is all about genetics and genomics. It's part of every pharma in one way or another, whether they were built on it or not. Most every rare disease company, of which there are many, are built on these discoveries. So from the biomedical space, we're already, I think, creating that economy, both on tools and platforms and on the applications to change humanity in a way. I haven't seen as much success in consumer. I mean, we all know about 23andMe. That was ahead of its time a little bit, but a very interesting, ahead in a business model sense, challenging business model, but great use of the technology and the tools in front of us. I think there'll be many more where these type of applications come forward. There is a fear we all have to watch out for, which has been around genomics for a long time, and that's the fear of, or the concern of, eugenics and improving humanity in ways that are not ethical or moral. And the tools are there. Right. They're so powerful now, they can be used and they can be misused. Right. And from a society perspective, we got to be careful on those type of things. Right. But what great applications we have in front of us to use them wisely.
Yeah, true. Well, looking at Jack's near-term future, we've just talked about the 6 sectors we see in the near-term future. What does the next 3 to 5 years look like for Jack's?
Yeah, I really want Jax to execute on this cells plus mice underpinned by AI link and become the partner for all of it, for all the pharma. And there may be others, and we'll welcome others. We would like to see the field advance to its maximum potential, however we get there. I think Jax is in a pretty uniquely powerful position because of our history with the mouse. And no one else has that history. So bringing that to cells and enabling and fostering and accelerating all of the discoveries that we have to get them through that valley of death and into translation. That's where I would like to see JAX. So when you think about, oh, we're doing drug discovery and we're moving forward, we're just part of that conversation for everyone.
So these new tools that you're gaining through NICEF, I mean, how's that going to show up in for JAX in the next 3 to 5 years? What will change in your laboratories, or won't your labs change too much?
Most of the scientists in JAX and externally are already using both of these types of tools for different applications.
Yeah.
What we haven't done is put them together. And I think this is classically one of those, the whole is greater than the sum of the parts.
Right.
And we bring them together. So imagine you're in big pharma and you're doing a study, and you do a study on cells, and you get an answer and you're all excited about it. You've got a new molecule and it's going to do something great for heart disease. And then you test it in an animal model and you get a different answer. Which one do you believe? And that's the way it works. I've lived in that world. It works that way. And it's very, very frustrating because you spend a lot of money. That's often at a point where the spend goes from a couple of $1 million to tens to hundreds of millions as you move into clinical trials. And you've got conflicting pieces of information or incomplete information or things you don't really know what to do with. But what if we move to a world where they inform each other? And you do that cellular study and it says, this is what you should see in the mouse. So that if you don't see this, then that's an answer. And if you do see it, that's an answer too. That's the one you really want to see. And so they inform each other. Now that's a game changer for decision-making and investment. That's what we want to see happen.
Yeah.
So that they start informing each other. As the trajectories are right now, that's not going to happen because the mice are used over here, or all animal models are used, and then the cells are used for something else, and they don't really cross paths. So we want to get those paths intersecting and informing each other.
Yeah. Wow. Well, speaking of precision, that's a really great story of how of what this acquisition really means. So, Lon, when you and I first met, you said something that stayed with me, and I think you've kind of hinted at it in this interview as well, which is that you've had this front row seat to genomics for your whole career. And you've seen wave after wave of, this changes everything. And then, of course, that inflection point falls short or doesn't come and takes longer than expected. Sounds like you still feel that way today. Is that true? And if so, do you think that this opportunity with NICEF might be an inflection point for the industry as a whole?
I think where we are, maybe it's me getting older, but we've been disappointed a lot in genomics. We have, on one hand, had the most astounding discoveries about human biology and understanding the defects that make us interesting in the way that we are, or the changes that just make us differ from one another. That's been phenomenal, and it continues to be phenomenal. But translating that to practice hasn't been as successful as I think we all hoped. Not in the way that we hoped, anyway.
Yeah.
But now I feel like what is missing? At scale, we can sequence every human on the planet if we really wanted to, and we had the resources to do it. We can edit any piece of DNA in any way we want. In addition, ask what happens. We can put those same genes into animals and ask what happens down the road. We can screen drugs at a pace faster than ever before. It feels like we're not missing. The only thing we're really missing is execution. So that's where I do think AI can let's bring all these data sets together. They're disparate, they live differently, they answer different questions, they're not really built to inform one another. Maybe that is the time. So I do think it's not just cells and mice, but that's a big piece of it. Cells and mice, and the way we think about drug discovery and gene therapies and cell therapies, delivery to tissues, which has always been a challenge in drug discovery. Delivery. Can I get the drug? I may have a great drug, but can I get it where I want, when I want, at a dose that works and is not too much? We're getting better and better at that. So it feels like putting the pieces together is now the challenge, rather than what we used to say is, if we only had.
Right.
If we only could sequence DNA, or we only could edit DNA, or we have all those pieces now. So now it's a matter of execution.
So can I put you down as cautiously optimistic?
There you go. Yeah, more optimistic than I have ever been. I come out of statistical genetics, and the phenotype that was once described of my community was those who walk into the light and spread darkness. Because we're always the curmudgeons in the room. And so for me, saying we just need to execute is about as positive as I can be. And I know I speak for a lot of my colleagues as well.
Well, interesting. Well, I will share this observation with you from our vantage point at GMG. When we look at JAX and NICEF together, it really does resemble something close to an inflection point, one where JAX has created the missing infrastructure, if you will, the kind of full stack biology platform that spans, as you mentioned, the in vivo models, human stem cell systems, organoids, automation, and data all under one integrated roof. And gosh, I can't think of any other place in the world that has that kind of end-to-end capability. And now JAX does. So, just to let you know sort of what we're seeing from the outside.
Well, we appreciate that. And I really like the description of the full stack. That is a very nice descriptor for us. Thank you.
Well, wrapping it up now, if you could speak to future business partners, and you are, what's one development that they should keep their eye on when looking at the genomic space and opportunities to get involved?
Oh, that's a very good one. I want to, oh boy, so many. The technologies are evolving every day. So I would keep my eye on those, and I mean all the technologies for delivery and for sequencing and for editing and all of those things. Uh, are there major breakthroughs on those? I don't know. They always surprise us, but they're definitely moving incrementally. Where I want to see it really going is, what if we have, on a platform sense, more baby KJ type examples coming through? Because that was not an incremental move. That was a paradigm shift in getting a brand new investigational therapy into the patient that needed it. If we start seeing that, and we can put that in a commoditized sort of way, whatever platforms it takes, that's a game changer. That's a game changer for drug discovery all the way through. That's where I would watch that space. And where is it? Neurological? Is it cardiovascular? What areas? Liver? What areas are the first to really make headway there? And I think we will see headway there.
So you're talking speed from determining that there's a problem to finding the solution.
Yes, and not just a bit better speed, but vastly better speed and greater probability of success in getting there. That's what the field has always needed. And then what industry will want to do is turn that into, from occasions of very rare diseases, into greater applications, because the model of investment will require that the return on that investment will need greater patients. And I think that will drive that field forward in exactly the way that it should. And
Yeah,
very, very important.
I think consumers are asking for health span, for looking for ways to live better, live longer. And they're looking to consumer products to be able to deliver them some kind of a benefit, and looking backwards toward what's in their DNA and how that can be used to understand what they need as a consumer is how we're viewing or seeing this sort of genomics economy come forward. But I'd like to ask, as companies and investors and new partners show up and want to be involved with JAX, what's the best way for them to engage?
Oh, by all means, contact us. You can contact from top to bottom. Any of us. We're a very flat organization, so any of us, we have a good track record. We're very proud of it as being a partner of choice, and we do like to collaborate broadly across diseases, across industries. We're very welcome to the new technologies and the new players. I think there's room for everyone in this, and sometimes the best ideas come from outside. And so all of these new applications are of great interest to us, and we'd be more than happy to discuss them.
Well, that is a great way to end. This has been a phenomenal discussion, Lon. Thank you so much for joining us on the Helix Exchange. You've been listening to Genomics Media Group, your source for the business of genomics. Subscribe on YouTube or follow us on your favorite platform at Genomics Media for insights you won't find anywhere else. Until next time, I'm Meg Samick Smith, and this is the Helix Exchange.